So welcome everyone, and I'm happy to have many joined to today's webinar presented by SynBio Canada. SynBio Canada is a volunteer organization working on strengthening the community of synthetic biology in all fields in Canada. Today we have two speakers. I'm happy to have Dr. Stuart Smyth from the University of Saskatchewan, who's worked for over a decade on genetically modified crops for improving life quality and having like a greener future, or like ensuring more sustainability. Dr. Stuart will start his talk in a minute, and we'll have a period of questions or interactions afterward before the second speaker, Serge, takes the floor. So I'm going to mute everyone except Dr. Stuart, and thank you for presenting today again.
Well, thank you very much, Fatima, and thank you to you and SynBio Canada for the opportunity to participate, and I'm looking forward to this. So I want to take a few minutes and just give you a bit of an overview on where I see SynBio having a role and how that's going to impact with some of the DSI aspects around food security and ag sustainability in the coming decades. So I just want to acknowledge the partners to my enhancement chair. I hold the only D4 research chair in Canada, where I've got producer organizations, private companies, and public institutions providing funding to the research my team and I lead. So one of the things about food is that we quite often become quite fixated in ensuring that we don't want to see too much innovation in the food products we're consuming, because we like to stick to foods that we're familiar with. But it's important to remember that this note, we're going to need to produce as much food in the next 30 years as humanity's consumed in the last 50,000 years. So innovation in food and food science is going to be crucial. Now, at an international level, synthetic biology is being regulated through the Convention on Biological Diversity. And it's regrettable, but there's been a significant push over the last several years by a lot of environmental organizations to try and restrict the development and application of SynBio technologies as much as possible. The problem at the international level is that there's no international institution that has a mandate for governing synthetic biology. So the CBD is trying to push its way into this area, even though it doesn't have a mandate from any government in the world to regulate non-living materials. So this becomes a real problem at an international level, is that there's really no organization or institution capable of governing synthetic biology at an international level right now. So that, and the lack of a clear definition on what or what is not synthetic biology, is creating some problems when it comes to the approval or the risk assessment of some of the products, technologies, and processes that can fall within SynBio. So depending on who you're looking at or what governments are making rulings, there are a variety of technologies that can be included within synthetic biology. Some aspects of gene editing are being included as part of synthetic biology, particularly those that don't involve any foreign DNA being transferred and are simply within crop genome editing techniques that are being applied. The FAO has recently reported that over the past 5 or 6 years, food security is again on the rise. from the period of about 2005 to 2015 saw a significant decline in the global numbers of food security, but it's risen over the past several years. And one of the significant tools that can help alleviate food insecurity is the application of biotechnology to crop agriculture, which has been shown to increase yields by just over 20% over the 25 years that GM crops have been adopted by countries around the world. And another important application of biotech is the use of biofortification, where crop nutrients, crop vitamins can be enhanced, because the FAO has identified that in addition to the nearly 700 million people that are food insecure, there's an additional 1 billion people that don't receive enough nutrients on a daily basis either. So gene editing is also providing a lot of opportunities to deal with food security, but I think one of the more exciting areas is the ability of gene editing to work and make sure that crop varieties are capable of producing at a sustainable or increased level as climate conditions change in growing regions around the world. And I think these are some areas that I've seen research in over the last 5 or 6 years. And you can see the different crop commodities and the different nutritional enhancements that are being done there. And I think this is really where food security is going to take a significant improvement. Some of it will come from increased production in industrialized countries like Canada, the United States, Brazil, Argentina, Australia, and those surplus productions being internationally shipped. But to really get at the heart of a lot of food insecurity, it's going to require the adoption of innovative techniques and technologies into countries that are food insecure. And we've seen several examples within the last 12 to 18 months in African and Asian countries where they've simply said, you know what, if we're going to address food insecurity, we have to adopt the technologies. And a really great example of this was the commercialization and regulatory approval last fall of BT cowpeas in Nigeria. The Nigerian government has indicated that they're going to make enough seed available this spring for 10,000 farmers to be able to plant an insect-resistant variety of cowpeas, contributing significantly to food security for those farms and families that are able to have the opportunity to adopt this. So I think there can be a substantial two-pronged approach to improving food security. Certainly, industrial countries like ours need to make sure that we're as cutting edge as possible to ensure that our yields are as high, but we also need to ensure that technologies can be adopted and commercialized in developing countries that have the ability to slip seamlessly into their food systems, and people are able to adopt, say, a gene-edited variety of cowpea or sweet potatoes, or other crops that they're familiar with, they know how to cook, and it's not a commodity that's introduced from another part of the world. I think digital sequence information is going to have a significant impact on the ability of crops to enhance nutrition, to improve their ability to combat changes in climate conditions. And where I see a lot of the advantages coming for this is as plant genomes can be sequenced, big data analytics are going to allow for plant researchers to search multiple plant genomes, so they can search, a dozen different varieties of wheat. They can look at weedy relatives for drought tolerance genes or other abiotic or biotic stresses that are beneficial to those weedy relatives or wild types, for example. And so then these genes can be recreated within the existing wheat genome of the variety that's being targeted. And that's going to make for significant opportunities to address climate change and food insecurity. So some of the field trials that are being done are reporting evidence of yield increases of 20 to 50% in some examples. So of course that's going to be a little bit high, and it'll drop as it gets transferred to field conditions. But the preliminary evidence on this is certainly promising. And where I think a big part of this is that it's going to allow for researchers at public institutions to have access to these plant technologies that biotechnology really prevented. That when you look at the 29 or 30 different GM varieties or crops that have GM varieties, very few of those have been developed by public breeders, and allowing gene editing to be used by public sector plant breeders it is really going to be a game changer, because so much of biotechnology really hasn't been available to them. One of the questions that certainly comes up out of the discussions around DSI is whether the sequencing of a plant genome is traditional knowledge, and there's been a lot of discussion as to how to ensure that benefits of innovative technologies do flow back to traditional sources of traditional knowledge, and this is an ongoing challenge, and certainly one that is not going to be easily solved. And there have been a lot of efforts over the last 3 to 5 years, and progress is being made. Certainly Environment Canada has been working diligently on this file in the last couple of years, and NOI and several others that are part of this call today have participated in some of those discussions to help provide advice and guidance on these issues. One of the things that kind of amazes me is that much of the public believes that more regulation is better for society, without realizing that there's a cost to this. Right now, capital is the most mobile resource on the planet, and we've seen investments flowing out of Europe in the last 3 years, since they decided that they were going to regulate gene editing as the equivalent of GMOs. Hundreds of millions of dollars have been transferred out of the European Union, and they're being reinvested in Canada, the United States, Brazil, Argentina, Australia, Japan, lots of other countries around the world that have more efficient regulatory systems than what the European Union has. So the problem, though, in Canada is that we aren't recognized as being terribly efficient when it comes to regulatory competitiveness. You can see we're not well ranked in terms of our regulatory burden. One of the big problems is that while Canada is recognized as a top 10 or top 15 country globally when it comes to our ability to fund research and development, we have a significant lag when it comes to our ability to commercialize products from our research funding. And in fact, we're ranked around 30th when it comes to our ability to commercialize products from innovation. So there's a significant gap between what goes into our innovation pipelines and what comes out as products for us as individual consumers. I think a really important message that is starting to be increasingly communicated to Canadian citizens and people more globally is just how sustainable agriculture has become following the commercialization of GM crops. So one of my graduate students is doing a research project where we're surveying farmers with a large survey to better understand what their crop rotation practices were in the early 1990s prior to the commercialization of GM crops as a form of weed control, and their most recent crop rotations between 2016 and 2019. And you can see the absolute turnabout, with about 40% of land in Saskatchewan being zero or minimum tillage 25 to 30 years ago, and virtually no summer fallow or virtually no tillage practices exist within Saskatchewan anymore based on the research we've found. So this has significant sustainability benefits, reduced soil erosion and increased moisture conservation. Chemical use has increased because we've got more seeded acres in Saskatchewan now than we had 25 or 30 years ago, but the environmental impact of the chemicals that are being applied is significantly lower than what it was 25 and 30 years ago. So even though we apply more chemicals on a per acre basis, the environmental impact of those chemicals being applied is half of what it was previously. So again, strong contributions to sustainability. So how do we communicate this in a world which the World Economic Forum has identified that we're really in the midst of an infodemic, that there's so much misinformation or disinformation being circulated on a lot of social media platforms? How do consumers know and where do they go for trusted sources? A good example of this was the Canadian Institute for Food Security's report that came out in November that identified that about 90% of Canadians self-identified as saying they have a limited to low knowledge of how food, crops and food are produced in Canada. So a significant challenge is that with 9 out of 10 Canadians saying we really have very little knowledge about how food is produced, and so they see memes or content on social media that's deliberately designed to misinform them, whether it's about the production of food or the safety and efficacy of vaccines. It creates a real challenge within modern society as to who is a trusted source of information and where can consumers go to access information that is based on facts and evidence and not on a hidden agenda to try and elicit donations to particular special interest organizations. We've certainly seen a lot of backlash from some social media platforms around the anti-vax message, particularly related to the COVID vaccines. And so one of the things I've done through the establishment of my research chair is to set up a website, and you can see the link there. We put out blogs on a weekly basis, 10 months out of the year, that promote knowledge about agriculture, about sustainability, innovation, food. I publish blogs written by my students to provide their perspectives on these areas and topics. So if you haven't heard of this and are interested, please go and check it out. It's an excellent source, and if you have people that are saying, where do you go for trusted information? Feel free to flip them Safe Food as a source of information. So just to sort of quickly wrap this up, I think that over the coming decade, innovation within food and agriculture is going to be one of the most innovative sectors in the economy. To a large extent, digital technologies have entered virtually every other sector of the economy, and within the last 3 to 5 years, they've started to come into the agriculture sector. And I think now that they've entered, they're going to sweep across the industry at an unprecedented rate. And just to a large extent, if we jumped 10 years into the future, I think there's significant aspects of agriculture and food production that would just hardly be recognizable to us today. The challenge, though, is that activist organizations are spending hundreds of millions of dollars a year fighting innovation because they've got vested interests and are really opposed to seeing innovations within the crop production and food sectors. So to a large extent, the last bullet has been put out there by Tim Caulfield, a health law professor at the University of Alberta, challenging academics across the country. And that's not just professors, that's people that are postdocs, that are graduate students, saying when you see misinformation, it's our responsibility to try and correct that through providing a link or a source to say, no, here's a bit of information that counters that. And so I challenge everybody on the call, to when we see information that is incorrect, to do our best to provide a a reliable source of information. I try to do this through my Twitter account, and if you don't follow me, feel free to give me a follow. I try to do a lot of science communication. I know there's others on the call or on the platform here today that are involved in this. And Fatima, I turn things back to you, and thank you very much for the presentation.
Well, thank you. It was a very interesting presentation. I actually really enjoyed the last part where you mentioned like the awareness that people are raising more and more on the GM crops, because the tolerance pre-COVID have certainly changed. Like the last year wasn't the best year for everyone, but it has certainly impacted positively, at least the GM food and the people's trust in science. I didn't know about safe food before, like long ago, but I really like also the impact that it has on some people. Like people can go and read the post and discuss about it on social media. Do we have any questions for Dr. Smyth? If not, I can start. I have a very specific question because it interests me for the future. I know that in certain grant applications, there's usually a part where we need to discuss about how safe for the environment is the application we are applying for. So for GM crops, how do you usually like justify the fact that it will not affect the biodiversity or that it will be safe for the environment? I would like you to have your input on that.
Sure. I've spent a lot of time trying to figure out what the best answer to that question is, Fatima, and it's a great question. And what I've come to offer as a response to that is GM crops have been assessed for risk by governments in 70 or 71 different countries. So there's about 30 countries that produce GM crops and another 40 countries that import them either for human food consumption or livestock consumption. And so a risk assessment is done for those import purposes. So we've got about 70 governments that have done risk assessments for 25 years. And in those nearly 4,500 risk assessments, not one of them has concluded that GM crops offer a risk to the human health or the environment that differs from the risks of producing a non-GM crop. Crop production has an impact on the environment and food security or human health. We know that. It simply, the production of food will impact the environment. There's no way we can produce food without it impacting the environment. So we want to make sure that any technology we're commercializing is no riskier than what we're doing now. And that's what the risk assessments have found for 25 years, is that the risk of producing a GM crop is no different than the risk of producing a non-GM crop.
Thank you for that. Any other questions? Well, I have another one, but it's like more general. Have you seen a lot of work on CRISPR activated crops recently, or it's not that, it's not yet?
I did a survey of plant breeders in Canada in 2018, and at that time, I think about 70% said they were either using CRISPR in their labs, both public and private labs, to do proof of concept. So I suspect, that was 2.5 years ago, so it's probably gone up a little bit. So maybe 75 or 80% of plant breeders would be using it, certainly in the laboratories. But I couldn't speculate as to where those varieties might be in the development pipeline.
We have a difference. We have a question here. Is there a difference from Caleb? Caleb is asking, is there a difference in public perception for CRISPR modified crops versus directed evolution as a gene editing technique?
That's a good question. Some of the consumer research I've done suggests that consumers have are more receptive of gene editing but it has to be connected to what the benefit is. So we found in an example where we said that a gene-edited crop would reduce chemical use, the majority of respondents said they would buy that product if it was priced the same as other products on the store shelves. So I think gene editing that shows either environment sustainability benefits would be welcomed by consumers based on some of the research that I've seen, that I've done, but also evidence from other academics they're asking similar questions to consumers.
Christopher is saying, should the question of safety not be about the technology, but about the traits being engineered in?
Right. So I'm not sure if you heard, Christopher, but Health Canada has been sort of trying to provide more clarity about the novelty regarding gene editing. And there was an announcement that came out about 10 days ago that they're getting ready to provide a public document for consultation. And there was a webinar held by the Canada Grains Council on Friday, expecting that it could be even released this week by Health Canada. But what it was articulating was that as long as gene editing didn't involve a trait that didn't have a history of safe use, there would be no requirements for additional regulatory oversight. So you could speculate saying, okay, well, herbicide tolerance in canola or soybeans or corn in Canada has a 25-year history of safe use. So if you wanted to use gene editing to insert that into a new variety of canola, corn, or soybeans, that wouldn't require any additional risk assessment. But let's say you wanted to put herbicide tolerance into oats or barley where it doesn't have a history of safe use, then it might require a risk assessment.
Are there other selection techniques? Like I know that in other organisms we do auxotrophic strains. Is that something that is like, is anyone working on something like that instead of working on herbicide resistance? Like can we change Basta for let's say planting the plants in the soil that is treated for not having, I don't know, not calcium element? Is that something that is possible?
I know there's research going on at my university that's looking at, the soil, the root health or the ability of roots to uptake, both moisture and nutrients. So I'm not a bench scientist, Fatima. I'm a policy guy. So a lot of your, the technical aspects of that, I simply can't respond to. But I do know that there is research in those areas that if it's going on here, I suspect it's going on at other universities across the country. And in fact, Trevor's probably the expert that that question could be directed to.
Great. Well, Trevor is asking if, I think it's a guess, what will be the first gene-edited, sorry, I missed that. The first gene-edited crop that is non-GMO label.
So in the States, CSIPC has non-GM canola that's been commercialized. Calyx has non-GM soybeans that have been developed through gene editing. They've been approved for import into Canada. Perhaps the first one that might be available would be the Simplot potato that's been developed for reduced acrylamide content. So Simplot was saying that they thought those potatoes would be available last year for production, but I haven't heard any subsequent information as to whether those varieties were available in the Maritimes or not.
Great, thank you. Caleb have suggested that we shift that to another panel, because I think it's a very interesting discussion for everyone. Thank you very much, Dr. Stuart. Today presentation was very interesting. I'm happy to share the Safe Food link because I got interested in it. I think many people can use that to share safe and complete information to fight over the misinformations on the social media. With that, I would like to thank you very much, and I think that will not be our last discussion about GM crops and the social awareness about it. And now, without more ado, I will switch. I will switch to Serge Nouemssi, our second speaker. Serge is a PhD candidate in the University of Quebec at Trois-Rivières, UQTR, and he's working on modifying microalgae for the green microalgae, more specifically for the production of bioactive molecules and food or nutrients. Serge will talk about the challenges he's finding, and we encourage students like Serge to share their aspects or their research projects to SynBio and be our next speakers. Serge, the floor is yours.
Thank you, Fatima. Thank you very much.
L'eau acceptable, je l'ai proprement, je l'ai plusieurs fois.
Okay, good afternoon, everyone. Thank you, Fatima, for this brief introduction. So today I'm going to present to you about my project, not my project, what I'm working on. So it's the production of bioactive molecules in Chlamydomonas reinhardtii, who is a microalgae that has been used a long time ago for the synthetic biology. So my presentation outline will be, I will present briefly a context on which we are now, and reinhardtii has a sustainable solution for the future, and the bioengineering of reinhardtii and the related challenges. And then I will briefly show the conclusion that I made for this presentation. So first of all, the world population is keep growing and expected to reach 9.7 billion people in 2015. And as this curve can show, we see that in 19, the population is keeping growing, and here where we are in 2020.
Serge, and excuse me, can you share in diaporama mode?
Ah, okay, sorry. Just a few minutes, sorry for that. Just 1 second, it's okay now? Good.
Yes.
So, and here where we are now in 2020, we see population is keep growing. So beside this growing of population, there is a impact factor that affects the environment it's a global threat. So how climate change can affect food security. So climate change, some statistics show that the climate change will affect food security in maybe 10 years. And in some parts of the world, we can see that treatment affects principally some food, some crop production like maize, wheat, and also pasture production. Here we can see that this production is decreasing during the time affecting by the climate change. And for that, the land scale production of crops will be really difficult in the future. That's why it's important to think to find an alternative that could help people to make food in the safe way. So a best solution, a sustainable solution that maybe we should think about now is it's a microalgae. So one of microalgae that we are thinking about is Chlamydomonas reinhardtii. And the phylogeny show that the Chlamydomonas reinhardtii is really close to the plant family. And this microalgae is an eukaryote photosynthetic microalgae, and the trigenome, like chloroplastic, nuclear, and mitochondrial genome are all sequenced, and we have lots of tools for his genetic engineering available. The transformation of genetic of Chlamydomonas reinhardtii is rapid, and the stability of we have also the stability of cell line. And then the cultivation of microalgae is really less cost, and it's practical. But some studies show that the Chlamydomonas reinhardtii have been used Chlamydomonas reinhardtii for the production as model for the production of biofuel and bioproducts. And also Chlamydomonas reinhardtii have been reported as industrial, has been established as an industrial host for the biotechnology. To engineer, there is a lot of nutraceutical pigment produced by natural products by this algae. So we can find like it's a source of pigment and fatty acid, and these molecules are really important for the health and for the nutrition supplement. And it's important for the health show that make microalgae can, with the nutraceutical that is produced, like a vitamin and lipids produced by this microalgae, we can consume it, and this will have an important role in the health protection in different parts, and also they can act like antioxidant, anticoagulant, and anti-cancer, and also can be an immuno-enhancer for the organism. So to produce this molecule in Chlamydomonas reinhardtii, some companies showed has already used Chlamydomonas reinhardtii to produce some edible, has used Chlamydomonas reinhardtii as like algae-based food production, and they show that, and the use of Chlamydomonas reinhardtii as edible microalgae have been approved by the Food and Drug Association Administration, like a general, like grass. And the company, one company named Triton, has used microalgae to modify, engineer microalgae Chlamydomonas reinhardtii to produce edible powder of this microalgae, rich in vitamins, vitamin A, protein, and a source of lots of lipids like omega 3, 6, and 9 fatty acids. So how to engineer microalgae to produce all these molecules? And recently, the recent project using microalgae is to produce mammalian breast milk in Chlamydomonas reinhardtii. And Chlamydomonas reinhardtii is used as an expression for the expression of recombinant protein. One of the example here is the lactoferrin in that microalgae, who is edible. So to engineer microalgae, lots of tools are available for this purpose. So we have these tools where we can find easily the sequence and blast them or align them to to verify our sequence of interest. And after that, we can find the strand of algae that we want to work with in the database, in the Chlamydomonas Resource Center, depending on what we want to do and the type of the transformation that the method of transformation that we are going to use. And after that, we can also engineer our gene of interest using this software named SnapGene. And there is a lot of software and tools available to engineer your sequence. First, we need to make the genetic construction, to design our genetic construct. So the choice of the gene of interest. So depend on what you want to produce in Chlamydomonas reinhardtii. You have to choose your interest gene and codon optimize it for the best expression in Chlamydomonas. Once the gene of interest is chosen, you now look for the reporter genes or tags. The role of this one is to make sure that after expression, you can easily detect your protein by using some techniques, biomolecular techniques to identify them by, if you are using like fluorescent protein, you can do it directly by the confocal microscopy to find out if your protein is expressed or not. And with the tag, you can do it by using Western blot, and after that, you will make sure that, okay, my protein is present. After choosing your reporter gene or your corresponding tag, you can also, depending if you want to make the construction with 2 different genes or 1 gene, it depends. You can use the flexible linker or a cleavage linker. And one of the example of cleavage linker that we have is the foot mouse disease virus 2A. That is autoclave itself during the translation of the proteins. After doing this, you need to add the regulator element in your gene, because the gene, for the expression, gene need to have like the regulator elements such as like a promoter who will help your gene to be transcribed and at the end translate. And you need at the end 1 terminator that will make sure that we say that, okay, the transcription is finished, so we can stop here. So, and at the end, you will choose the choice of your vector and the design. So this shows all the elements that are already listed before to show how to design your vector. So your vector needs to have the reporter and regulator elements and the ribosome binding site. Then you have your gene of interest. Then you can which you can clone in the multiple cloning sites or depending on what you want to do, the restriction enzyme that you are going to use. And at the end, once the vector is designed with your insert, you have to clone it, use the method that you want, and it depends on what you want to do. And there exists a sort of method to clone the gene of interest into the expression vector. So, and after that, we can transform after cloning, the vector can be transformed with your insert, can be transformed in the microRNA using different methods that are available for this strain, for this microorganism, by electroporation or glass bead method or biolistic or agrobacterium mediated transformation. And after that, we need to screen the transformant that we have. Here is the challenging part working with Chlamydomonas reinhardtii. The first step of the screening method is the subculturing of the of different transformants in the selecting media. So here we are subculturing our transformant in the selecting media containing the desired antibiotic, the resistant marker that we use, that we choose for the cloning, for the expression of our transgene. And then we can do the colony PCR to make sure that our DNA sequence is present or not. And this, it has been reported by recently that Chlamydomonas, for the expression, to make sure that we have to expect it to have one good transgene expression in Chlamydomonas, we need to at least select 100 plants harboring the full cassette, so harboring the full gene of interest that we we inserted in the algae. And depending on the method that we are using, we can have like 1% or 2% of the expression of the expressed clone. And it has been also shown that there is a mechanism, the mechanism called gene silencing that occur that is highly present in Chlamydomonas genome. So these can also affect the expression of the gene of interest in Chlamydomonas reinhardtii systems. Behind all this, we are in our lab working with Chlamydomonas was a big challenge, and we think that developing a method to easily screen lots of clones that we obtain after transformation can be a best thing to work with this strain. So in this, this paper has been published in September 2020, and here we show the different, how can we, you can easily screen your strain after transformation. And for that, we make a layer of, we were striking our transformer based on the layer of 96 well plate and use the multi-channel pipette to like pick them, how to pick them to do the genomic DNA extraction. And then we are using the same system to make the PCR product loading in the gel electrophoresis. And after, and then we can, after that, select all the clone that harboring the cassette, the gene of our gene of interest. And after this, you need to go to the step of the protein to see if you are using, depending on the selection marker that you are using, if you are using the fluorescent protein, you can easily find out if your gene is expressed or not using the confocal microscopy to check if there is fluorescent. This image shows that here is the wild type strain, and here is the transformer harboring the M-venus fluorescent protein. And the brightness of this strain show that, of this clone, show that this clone is positive for the transgene that have been inserted in the Chlamydomonas. And after that, if you don't use the fluorescent protein, you can directly go to the protein detection by Western blot. And here, depending on what you did, depending on the size of your protein, you can easily find out if your protein is expressed or not. And it has been shown that there's a high level of glycosylation in Chlamydomonas genome. And this also can be a big challenge for the Chlamydomonas screening by the Western blot method. And also we can also use the RNA expression or quantification by real-time PCR or by real-time PCR or qPCR to know if our messenger is present or not. So, and at the end, after all these steps that we passing through, you can then now analyze if your metabolite is present or not by using the HPLC, GCMS, or UPLC, depending on the retention time, and using some standard that you will, some standard that are corresponding to your desired, to your gene of interest, to your specific metabolite that you want. And based on the retention time of each standard, and you can easily find out if your metabolite is produced by the algae or not. So this is actually the how you can engineer microalgae and how you can produce the metabolite of food in the this strain. And also there's another method where you can also not to like modify, like insert a foreign gene in the strain, but just modify some pathway or for the redirection of the metabolism. If, for example, for the high amount of lipid production, we can just make the modification by using the CRISPR-Cas that will help you to maybe knock out some gene or knock in to like enhance the production of the desired metabolite in one direction. And we have been also published a paper that step out all the tools and steps that you can use for the engineering using this CRISPR-Cas system to engineer Chlamydomonas reinhardtii. And after all the steps that we highlighted, you can now use the strains that are positive for your transient expression to scale up for the production. And here is like a summary of all the things that I talk about. So first you need to engineer your strain, and after that you need to do the high throughput screening selection of the strain that you transform, and then you can process for the development. And after this, you will have like the product that you need in the high scale, but in the low cost. So in conclusion, why Chlamydomonas is a good organism to engineer? Because Chlamydomonas has a good, is a organism really close to the plant, and there's like this microalgae have like a similar system to the plant with the compartmentation of different of the organelles present in this of the microalgae. And it's also really easy to produce in the lab scale or in the industrial scale using only the bioreactor. So we don't need land scale to produce microalgae. Why Chlamydomonas is challenging? The big challenge here is that the gene insertion can be done real easy, but knowing where it got inserted and its fate is the tricky part. The challenge of using Chlamydomonas has like a biotechnological host can be overcome using the biotechnology. So we think that Chlamydomonas can be the hub for the future to have like a safe food and also a green world, because this algae is a photosynthetic and can only use the light source to produce, to give us the product that we want. So I would like to thank my director and co-director, Professor Isabel and Professor Hugo, and also the company for which I'm working during my PhD, and the founder that allow us to work in this in our project in the lab. And also I would like to thank all my colleagues, and I'm so glad to be a part of this lab, and we are sharing a lot of knowledge during this few years that I'm working with them. So thank you.
Thank you very much, Serge. That was interesting. I think we have few algae people on the chat. Who wants to start? Caleb, we have a question from Caleb. Did you protoplast your cells prior to electroporation? Do you do enzyme cell wall degradation before you do the electroporation? And also, do you knock out the nitrate reductase gene and use potassium chlorate to select out non-edited strains? It will make your life much easier. That's a suggestion for you. The edited strains will grow on potassium chlorate. So would you like to answer the first question? Do you protoplast your cells?
Thank you for the question. For the first question, we are using a wild type strain, so we are not protoplast our cells before electroporation. So we use the wild type and directly electroporated. So we don't know if, but we are having a high efficiency of transformation because we are having lots of clones, thousands of clones, and I don't know if the protoplast can help for the protoplast degradation can help for the insertion of the transgene in the better way.
You can try it. We have 2 other questions. I'll start with the short one from John Clark. Sorry, can you grow Chlamydomonas heterotrophically?
Yeah, of course. That's the advantage to work with the Chlamydomonas.
So if so, what's the highest cell density of phototrophic versus heterotrophic?
Sorry?
What's the highest cell density, phototrophic versus heterotrophic? Do you have biomass when you grow heterotrophically? I imagine you do.
Yeah, exactly. But there's not like a big difference between the two way how you are growing it autotrophically or heterotrophically. So we are growing algae actually in the simple media, which is really cheap. It's a tap media with only salt, and also you can also put microalgae. You don't need like carbon source, a lot of carbon source to grow. So heterotrophically or autotrophically, I think we should have the same biomass at the end.
Comparing the fees of the supplementation, I guess, but I think there's many not very expensive supplements like carbonates or
exactly.
Okay, we have another question from James. Did a scaling project on a GMO strain of Chlamydomonas in 1,000 liter photobioreactor tracked and established that the genetic modification held? The targeted compound was a therapeutic protein. Well, I didn't get.
Sorry, there was no question there. It was just more making a statement because a lot of these discussions are around, looking at the opportunity around scaling. And I spent the last 15 years growing genetically modified and more natural-based algal strains. So just kind of putting it out there that there is some people that are looking at the scale-up side of this because a lot of the people doing the work under the hood don't. The scale-up component when you get into CIP and things like that becomes quite complex. And just let you know that there is industry out there that have spent the last 15 years studying this space for this type of opportunity. So anyway, that was what that was about. It was more of a statement, not a question. Great work that you're doing, by the way, Serge.
Thank you, John. Well, I think Serge, he's doing the modification, and I think that like the scale up could be usually in such projects, scale up is held by other companies or collaborators. Yeah, like mainly because like in our case or in Serge's case, the lab space is limited, so we don't have photobioreactors of big volumes. That could be a limiting factor for people who actually do the transformation versus the people who grow them for the scale up. But that's, yeah, I've seen some production in Chlamydomonas. It sounds interesting. Any other questions for Serge? We welcome also French questions in French. I can translate. Well, it's 2 o'clock, so if there's no more questions, we can stop here. I would like to thank all of the people on the chat and all of the attendees. I would like to thank our 2 speakers, Stuart and Serge. It was very interesting to hear you both. The very different works, but they all lead to food safety and food security in a GM aspect. I think myself, I enjoyed both of your presentations and the discussion that was afterward. Thank you very much.
Thank you for the invitation.
Thank you. And if anyone is interested, you can continue the discussion on the SynBio Canada Twitter account. You're welcome to add your comments, your questions, and output. Thank you, everyone, and have a great day. Enjoy the spring. The spring is here finally, so enjoy it. Chat. Great. Okay, I'm going to close the... Merci, Serge. Bye, everyone.
Bye-bye.
Yeah, that's for sure. We need more policy advocates here.
Félicitations, Serge.
Je pense qu'il n'est plus là, Amadou.
Ah, ok. Félicitations, Patrick. Ça a très bien réussi. Félicitations.
Merci. Bye.
Ouais.